So this is where we left off on, is that just Monday? Yeah, Monday. This is where we left off on Monday. And are there any questions on this? So those of you, there's 2 of you in HWS in sports nutrition right now, and there's several other people that have taken sports nutrition. And this figure goes really well with the protein section in sports nutrition. Because one of the things we talk about with individual amino acids like leucine or getting essential amino acids in the post-exercise state and getting your protein drink after every workout is twofold. The first is that you want to stimulate protein synthesis above if you had just not done anything, right? But it's important to show that in response to resistance training, we do have some protein synthesis going on, right? It's not like you get nothing. It just doesn't, it's just that the protein breakdown, especially if in a high intensity workout, can often outweigh the amount of protein synthesis that's there to make up for it. And you end up with a net loss or a net negative protein balance. So those of you that took HWS325, remember we read that paper by those protein guys, Tipton and Ferrando. And Tipton and Ferrando they were the ones that have done the tracer studies and established that we get benefit on a per workout basis. So there were a couple different sort of hypotheses in regards to response to training. One of them was that protein breakdown gets less as training goes up because you become more efficient and more resistant to the stress of training, right? So if protein synthesis stayed the same and protein breakdown went down, then your net balance would go up, right? The other theory was that as we train on a regular basis, we get more protein synthesis because we become more responsive to that training input. Protein breakdown stays the same and we get a greater balance. Right? But what they demonstrated was basically protein breakdown stays the same and protein synthesis stays the same. And we get a benefit from, or we have an opportunity to get a net positive protein balance on a per workout basis. So every workout has a purpose, even recovery workouts. And this is something that I come across a lot when I'm working with elite athletes. They think, well, going for like a 2 or 3 hour bike ride at a really low intensity conversation pace, if you will, that's barely doing anything, right? So why would I need like a full protein shake? But the reality is that's still different from just sitting, right? There's still stimulus being put on the system, even if it's a low intensity stimulus, right? And you want to get the adaptation to that. So things we've talked about a couple different times. There's just a guy in a really scary like skull mask running through the middle of campus. Caught my eye. He had khaki pants on, a normal shirt, and like a really scary skull mask on, and then he was pretty interesting. Okay, so the point here is that we've talked about even for power athletes, especially from a recovery standpoint, having some aerobic fitness is important because it's going to help you. And some of that aerobic fitness can come from really low intensity aerobic exercise to get some of those adaptations and angiogenesis and increased mitochondria and turning on PGC1 alpha and those types of things. It doesn't always take super intense activity to do that, right? So 2-3 hour hike on the weekends and then a recovery drink. Those are the types of things that you can take advantage of. Right? And depending on how you program it, you can try and minimize the effect that sort of endurance training has on your peak power. Right? So there's a lot of things to kind of juggle there. But this is really important. So essential amino acids suppress protein breakdown, stimulate protein synthesis, and the combination of that leads to the highest net protein balance. And this was demonstrated by our protein boy Tipton. That's what the article in 325 is about. And so this is important information. Questions on this? Thoughts on this? Okay, so what then are the mechanisms? So it's one thing to say, well, we want to get positive protein balance so we can get them gains, but it's another thing to say what are the intracellular signaling molecules that actually are important for distributing that signal and getting those gains. So there's three main things that we're going to talk about in this class. Now there are other factors, right? But as I've said many, many times, what we're trying to do is break down examples of intracellular signaling that are well established as being integral in conveying the adaptations to the specific situations that we're talking about. But there's always new stuff that's being discovered, and there's new ways to, just for example, in response to this, we were in HWS325, we were talking about how certain specific amino acids, when taken in concentrated doses, can have beneficial effects. So we see things like branched chain amino acids, or particularly leucine. Supplementation with it has been suggested in a variety of different things. And there's a supplement which is a metabolite of leucine called beta-hydroxymethylbutyrate. It's a supplement that's sold called HMB. And just this week, there were 2 or 3 new articles that were published talking about, there was one study in animals that was studying whether or not HMB was toxic in different ways. And then there were 2 other studies that demonstrated there was a suppression of protein breakdown when people were taking HMB during training. And if you can suppress protein breakdown, even if protein synthesis is the same, you can maximize this net difference. Does that make sense? So it's not always about just turning on protein synthesis or stimulating anabolic signaling. There's also an important factor to be played with suppressing breakdown and preserving tissue, so your net balances. So there's always stuff that's coming out in regards to new information about molecular mechanisms and supplementation and training protocols and crosstalk between training and stuff like that. We're just talking about some examples so you can get the language down and some of the understanding. So if you ask yourself, well, why is HMB, for example, causing to suppress protein breakdown? I wonder which molecular mechanism that is. Is it stimulating mTOR? Is it suppressing myostatin? Et cetera, et cetera. Right? So we're just going to use these 3 examples. The first is mTOR. This is a signal transduction pathway that increases protein synthesis. We have myostatin SMAD pathway, so we're going to talk about this. Myostatin is actually a brake on skeletal muscle synthesis, so we would want to inhibit that if we wanted increased skeletal muscle. And we'll talk about myostatin knockout animals. So have you guys ever seen that super jacked bull? You know what I'm talking about? What's that? Well, I showed you in EXS323, right? Pretty sure. Oh, in this class I did? Okay. So that's a myostatin knockout animal. And then we're also going to talk about satellite cells, which are these undifferentiated cells that I introduced a couple days ago, that kind of, they're pre-muscle cells that are in the, what's called the satellite cell niche or stem cell niche. They kind of reside in the shadows, and when they're needed, they're given some signals and they can turn into new cells, right? What? Mm-hmm. Exactly. You just need a stimulus, you need a bat light, and then the satellite cells are activated. Okay, so one of the things that I want to stress is how complicated, so this is a screenshot from my favorite website, cellsignaling.com. And I want to just stress how kind of many different things that mTOR can regulate. So mTOR is, the name for mTOR comes from its sensitivity to a toxin called rapamycin, or a drug called rapamycin. So mTOR stands for mammalian target of rapamycin. Rapamycin is this naturally occurring drug. It's widely used to manipulate mTOR within cell systems in animals, and to look at the effects, the downstream, both upstream and downstream of mTOR signaling. So this is a classic molecular biology technique where you apply some sort of stimulus or diet or training session or something like that, and then you can measure the activation of mTOR. You can measure the actual mTOR complex, and you can measure its activation by measuring the complex, but oftentimes what you'll do to measure mTOR activation is you'll measure AKT above, upstream, and then you'll measure the phosphorylation of either 4E-BP1 or P70S6 kinase. These are direct downstream targets. So if we measure through Western blotting the phosphorylation of 4E-BP1 or P70S6 kinase, we can use that as a surrogate for mTOR activation. That makes sense? So, for example, we see this is activated by growth factors, hormones, cytokines. What is an anabolic hormone that we talk about a lot? Natalie, what's an anabolic hormone that we talk about a lot? Stimulated by glucose, rise in blood glucose. What? So insulin is a growth factor, right? Insulin binds to the insulin receptor. We know that insulin activates PI3 kinase and activates AKT. AKT then activates mTOR. So one of the things that you can do, for example, is if I have some cells in cell culture, and then I collect those cells and measure their level of AKT activation and their level of P70S6 kinase, and then I add, for example, insulin, and then scrape up those cells and measure the activation, I'll see AKT turned on and P70S6 kinase turned on, and then I can assume that mTOR is activated. Okay? And then the other thing I can do is take these cells, add insulin to them, and add rapamycin, and I would get no activation of, AKT because it's upstream of rapamycin, but I would get no activation of P70S6 kinase. Do you understand? So this is how we can understand where a drug like rapamycin affects a pathway. So if I give insulin, and rapamycin, everything up to here is going to occur. But because rapamycin blocks the activation of mTOR, then anything downstream of mTOR is going to be prevented. And this is how you can unpack cellular signaling pathways, is by manipulating things at different places, either genetically or through drugs, and then stimulating them or putting them under different conditions. So mTOR and rapamycin are near and dear to my heart. The paper that I published, that was the main paper I published for my PhD, was about mTOR and the regulation, a new upstream regulator of mTOR in response to feeding. So we know that feeding activates mTOR, and then mTOR activated signaling molecules in a particular order, and we didn't know that. That's what I identified as a PhD student. So basically, mTOR is really important for turning on protein synthesis in the post-exercise state. And mTOR is really important for the adaptations and our gains from training. Okay? And what's interesting about it is that mTOR is not necessarily related to turning on, it's related specifically to some types of exercise, but for the most part, it's the exercise stimulus that the cell senses, and then mTOR is turning on general protein synthesis. Does that make sense? So the signals are going to activate different transcription factors and be sensed by the cell in different ways that are going to turn on certain transcription factors that lead to the expression of what? What do transcription factors turn on the expression of? What? Generally. Transcription factors enter the nucleus and turn on the expression of what? Say? Genes, right? And depending on the stimulus, there's different activation of different transcription factors that turn on the expression of different genes, right? What is one of those that we talked about a lot in the endurance section? What? So, no. What was the one we talked about where we showed the image in real time of the transcription factor in the cytosol, and then there was an increase in calcium and it entered the nucleus? No, calcineurin was the signaling molecule that activated what transcription factor? NFAT. NFAT was activated and goes into the nucleus and turns on the expression of genes, right? Those genes getting turned on make what? What is gene expression? What is it measured by? What? What does gene expression make? Protein synthesis is a process, not a product. So transcription factors bind to certain genes, and then what do they produce? What leaves the nucleus so that it can be what? mRNA, right? So gene expression is measured by mRNA. Now, to get that mRNA into protein, we have to turn on what? What do we do with a strip of mRNA? Is mRNA protein? No. So mRNA has to be what? Translated into, well, it's already RNA, and it gets translated into a protein, right? Protein synthesis. So turning on mTOR, which turns on protein synthesis, is occurring simultaneously to changes in transcription factors and increases in mRNA and increases in gene expression, right? Because you can turn on all the gene expression that you want, but if protein synthesis isn't there, you can't make that protein and then get that change in phenotype or get that adaptation, right? So this is another piece of the puzzle that's occurring. So when NFAT is entering the nucleus and turning on transcription and producing mRNA, and that mRNA is going to the cytosol, if that mRNA gets to the cytosol and the ribosomes aren't activated and there's no protein synthesis, that's an issue, right? Then you can't turn that into a functional protein. So when we're talking about protein synthesis in the post-exercise state, we're talking about turning on the process that takes advantage of the stimulus you just placed on the system that led to differences in gene expression, and then you can actually turn those genes that were turned on into functional proteins that give you an actual adaptation to that training session. Does this make sense? All right? And that's why, like, when you do different types of exercise, it's unique stimulus that turns on different genes. But for every type of exercise, we need to turn on protein synthesis so that we can turn the genes that were turned on into actual proteins and get the adaptation. Does that make sense? And there's different sets of genes that are turned on during different types of exercises because there's different types of signals that are stimulated during different types of exercise. Right? So when we do endurance training, there's more calcium in the system, which is a different signal. It turns on NFAT to a different degree. NFAT turns on gene expression and codes for genes that are specific to endurance training. Not only that, but the introns that get spliced out of the genes that NFAT makes turn off gene expression for type 2 fibers and type 2 characteristics. So now we have all this mRNA in the system, but we have to convert it into usable protein. And the same goes when we do resistance training. So we stimulate the system in a specific way. It senses load, it senses nutrition, it senses intensity, it senses hypoxia, it senses all these different things that we're inputting into the system, activates all these upstream characteristics, turns on unique genes, but those genes have to be turned into protein. And in the case of resistance training, if you're trying to, say, add muscle mass, then you're going to stimulate the gene in a way that leads to an increase in cross-sectional area of the myosin heavy chain, right? But you have to have protein synthesis to actually carry that out. And one of the big factors that regulates whether or not protein synthesis occurs is the activation of mTOR. But look at what else mTOR is related to. Do any of these seem familiar? Hypoxia-inducible factor 1. That was one of the ones we talked about during the angiogenesis section, right? That turns on VEGF and angiogenesis. That was a big one that we talked about, right? Another one that we talked about was PGC1 alpha, which leads to mitochondrial metabolism, right? We've also talked about what are called eukaryotic initiation factors. Eukaryotic initiation factors like 4EBP1 and EIF4G lead to mRNA translation, which leads to proliferation. We want proliferation and cell growth in the post-exercise state because we're trying to synthesize the proteins for the genes that were turned on in response to the training stimulus we just applied to the system. Do you understand? So mTOR and what's interesting is there's two mTOR or mTORC. The C stands for complex. And the complex is based on the fact that mTOR is only activated if all these other proteins, the raptor protein, the deptor protein, and the G beta L protein, can combine with the mTOR protein to make this mTORC1 complex. That's how it's activated. Right? mTORC2 has these other SYN1 and PRR gamma linked to the Richter protein, and that's called mTOR complex 2. And what's interesting is mTOR complex 2 turns on AKT, which turns on mTOR complex 1. And so one of the things that's thought is that like resistance training itself is sensed by mTORC2, which turns on AKT, which turns on mTOR. So you get some benefit even if you don't have an anabolic growth signal. Right? Protein synthesis is turned on even if we're in the fasted state. But we get more activation when we give it essential amino acids, and we get even more activation when we combine it with glucose that releases insulin. And that's because we can get more mTOR activation if we activate it in multiple different independent pathways. Do you understand? That's how we maximize response to a training stimulus. So you apply the stimulus, which is sensed by the cell, which activates certain transcription factors that lead to certain gene expression profile. But you have to make those genes into proteins by turning on protein synthesis. Protein synthesis is turned on through mTOR, and mTOR is regulated by multiple different pathways. So how do we know that mTOR is regulated, and what is the role of mTOR activation in the post-exercise state? If we take at rest an individual and then they do a resistance training exercise, we get a significant increase in muscle protein synthesis. If we give that organism rapamycin and put them through the same amount of exercise, you get no increase, post-exercise increase in protein synthesis. Super powerful, right? Rapamycin is a really powerful anti-anabolic drug. There's a lot of interest in rapamycin as a drug treatment for things like cancer, for example. Because if cancer mutations turn on mTOR, if we can block mTOR, then it could be a way to slow down growth. So that's just one of these implications, right? But in the sense of getting adaptation in response to exercise, you want to be able to turn mTOR on. Does that make sense? One of the ways that you can turn mTOR on, one is through training itself. We see that here. And then there's other specific amino acids and nutrition signals that can also turn on mTOR. Okay? So let's just unpack this. You good, Nico? So we've talked about all this stuff. All right? We have growth hormone, which can activate IGF-1 or insulin. What else can it activate insulin? So a rise in what? A rise in blood glucose level also releases insulin, right? Now, we often put our blinders on and we think of insulin as only being this facilitator of skeletal muscle glucose uptake. And we ignore all these other really important things that insulin does, like turn on protein synthesis, turn on anabolism, turn on, suppress fat breakdown, promote fat storage, turn on glycogen synthesis, anabolic processes, right? Building up processes that are associated with a high energy balance, so lots of energy in the system and low work being placed on the system. Does that make sense? All right. So we have a release of insulin that phosphorylates the insulin receptor. The insulin receptor binds insulin receptor substrate 1, that's IRS, and turns on PI3 kinase. PI3 kinase leads to phosphorylating this protein called PIP2, which stands for phosphoinositide phosphate 2, to phosphoinositide phosphate 3. PIP3 activates this membrane-bound protein called PDK1. PDK1 activates PKB and AKT through specific phosphorylation on a threonine and a serine residue. Right? Who's with me? So, and why this is important is because certain inhibitors and certain signals that turn off AKT or turn off IRS1 actually phosphorylate those proteins on different amino acid residues that suppress their activation. Right? So phosphorylation does not always mean activation. Phosphorylation on specific catalytic enzyme or catalytic amino acid residues will activate the protein. But on inhibitory residues, it'll suppress activation of the protein. But in this case, we activate AKT. Okay? So let's go back here. We have growth factors, which include insulin. Insulin activates what? IRS1. IRS1 leads to activation of PI3 kinase. PI3 kinase converts PIP2 to PIP3, which activates PDK1. PDK1 activates AKT, which is also known as PKB. AKT/PKB then can activate mTOR through the inhibition of TSC2. And this is one of the more straightforward cellular signaling pathways. All right, so look here, we have TSC2, which is inhibited. Now, under non-protein synthetic conditions, TSC2 is an inhibitor of mTOR. Okay? Because it inhibits this protein called Reb. Okay? So when you inhibit TSC2, which is an inhibitor, then you allow for the activation of Reb, and therefore Reb can activate mTOR. Let's go back here. Okay.
You deactivate it to reactivate it?
No, so this is when I knew I loved molecular biology. When you get the concept, if you inhibit an inhibitor, you activate it. You activate a downstream protein. Right? If you block an inhibitor, you activate the downstream protein. So TSC2 is an inhibitor of Reb, which is an activator of mTOR. And if you inhibit TSC2, then you can allow for the activation of Reb, which then allows for the activation of mTOR. And that's what we see here. So we have insulin, IRS1 and PI3 kinase, PIP2 to PIP3, activation of PDK. PDK turns on AKT. AKT inhibits TSC2. That allows for the activation of Reb. Reb then can activate mTORC1, and mTORC1 can turn on eukaryotic initiation factors and P7 and other growth signals like P70S6 kinase. Right? So we go here. We have insulin receptor, IRS1, PI3 kinase, PIP2 to PIP3, activation of PDK1, activation of AKT/PKB. This is related to mTORC2 signaling. That's up here, right? mTORC2 activates AKT as well. So then, but AKT, PKB, all right? So this is mTORC2 activating AKT/PKB, right? So PDK1 phosphorylates it on threonine 308, whereas mTORC2 phosphorylates it on serine 473. Now, activated AKT can block TSC2, which activates Reb, which activates mTOR1. And mTOR1 activates then 4E-BP1 and P70S6 kinase. 4E-BP1 is an inhibitor of eukaryotic initiation factor 4E. And so when we phosphorylate 4E-BP1, we relieve the inhibition and activate EIF4E. We turn on P70S6 kinase and look at what we get. Ribosome activation and mRNA being converted into a protein. Okay? And that's the same thing that we see here. Insulin to IRS1, PI3K, PIP2 to PIP3, AKT, mTORC is going to phosphorylate this AKT on serine 473. PDK is going to phosphorylate it on threonine 308. Then we get activation of AKT, inhibition of TSC2, activation of Reb, activation of mTOR. We get released inhibition of 4E-BP1, which is inhibited, allowing for the activation of EIF4E. And that leads to protein translation, mRNA translation. P70S6 kinase is also here and activates mRNA translation. The other thing is there's all these other eukaryotic initiation factors. EIF4G is just one example of that. All right? Stick with me, Trace. All right. So now, what can turn on this signal? Well, we've only talked about one of these things, which is what? Insulin. But Here we have weightlifting itself, resistance training itself has what are called mechanosensors, or things that sense mechanical work that directly inhibit Reb. When you inhibit Reb, then you inhibit an inhibitor and you turn on mTOR and you get some protein synthesis. Endurance exercise can also turn on mTOR. And endurance exercise turns on mTOR through the activation of what? AMPK. And how is AMPK activated? AMP kinase. It's activated by AMP. How is AMP made? Through what reaction? The two ADPs make an AMP and an ATP. What's that reaction called? Who said that? Say it. Yeah, the Lohmann reaction, right? So two ADPs make an AMP and an ATP. We get a rise in AMP, turns on AMP kinase. We know that AMP kinase leads to things like activation of AS160, which moves GLUT4 to the cell surface, remember? We talked about it being responsible for contraction-stimulated glucose uptake in the muscle. And then we also have AMPK stimulates glycogen breakdown. And that makes sense because we want to use glycogen when we're running, right? We also have increased calcium under these conditions. We have increased AMPK. AMPK can phosphorylate TSC2. TSC2 then is an inhibitor of Reb. It releases the inhibition of mTOR and it leads to mTOR activation. So do we need mTOR activation only after resistance training? No. We also need mTOR activation to maximize the adaptation to endurance training, right? Which is why post-exercise protein drink is really important even after you work out, even after you do your cardio. Because you're not trying to get big under those circumstances, and you don't have to worry about getting big under those circumstances because you haven't given the cell the signal to get big, right? AMPK and these other transcription factors like NFAT that are turned on during endurance training are specific to endurance training related gene expression. But you have to turn those things into protein synthesis. Too often we think of protein synthesis as only being related to muscle, that protein is equivalent to muscle. And it's not. All these proteins, glycolytic proteins, glycogen regulating proteins, all these different signals are things, angiogenesis, that lead to really important training adaptations. And so protein synthesis needs to be turned on regardless of if you do endurance training or weight training. And we can do it by stimulating insulin and turning on this signaling pathway and turning on mTOR and turning on protein synthesis. And finally, we have one last direct mechanism, which is amino acids themselves, particularly leucine. This is why I say leucine can turn on protein synthesis in an insulin-independent manner. Anybody that's had EXS323, HWS325 has heard me say that before. This is where that comes from. This is not related to insulin. It is amino acids directly inhibiting Reb, which releases Reb's inhibition of mTOR and activates protein synthesis, right? So if you don't want high insulin when you're sleeping overnight, this is why there's some suggestion that taking like 5 to 10 grams of leucine or 5 to 10 grams of branched-chain amino acids, of which leucine is one of them, before you go to bed, is a way to increase protein synthesis overnight without stimulating a big insulin response. Because we want to burn fat as an energy source overnight. We don't want to have you don't want to eat a bunch of ice cream and then go to bed. We don't want high insulin. Insulin is low. We want gluconeogenesis, we want fasting, but we also want protein synthesis for recovery under those conditions. And this is the basis of understanding those recommendations from a cellular adaptation standpoint. Okay? Cool. We'll redo this slide. We'll redo this slide on Friday, and then we'll move on to the next one.